Hey guys, welcome back. In today's session, I'll be discussing about the activities given in class 10 biology in chapter 1, that is nutrition. Here the first activity is presence of starch in leaves. Okay, so about this activity let's discuss. Take a leaf from a plant and take methylated spirit in a test tube, okay, and arrange the apparatus as shown here in the figure. This leaf should be soft and thin, okay, so soft leaf and the thin leaves that are well exposed to sunlight, okay, well exposed to sunlight. Such leaves must be selected, okay. So we have to take a leaf from a plant and that leaf must be soft, thin and well exposed to sunlight. Such leaves must be used for this experiment. Then take a methylated spirit in a test tube and arrange the apparatus as shown here, okay. You can see here this is a beaker, okay, and inside you will find boiling water. Then here the leaf is placed inside ethanol or methylated spirit, okay. A boiling tube is here and this is asbestos gas, and this is tripod stone, and here Bunsen burner. This is about the apparatus used for this experiment. Now here first boil the leaf in water and then transfer that leaf into a test tube containing methylated spirit, okay. So here the purpose of boiling the leaf is to remove the chlorophyll, okay. So first we have to boil the leaf in water and after that it is transferred into a test tube containing methylated spirit. Then boil this tube in water bath as it is shown here. The here test tube, inside this methylated spirit or alcohol is present and inside the leaf is placed and it is boiled in water bath, okay. So here the objective is to remove the chlorophyll from this, okay, or the remove the green color of this leaf, okay. Now after this chlorophyll, when it is boiled, okay, when the leaf is boiled in this water bath and the methylated spirit, okay, the chlorophyll dissolves. Chlorophyll gets dissolved, okay, in spirit, dissolved in spirit, okay, and the leaf becomes pale. The leaf color, it changes to pale, okay, due to the removal of chlorophyll from the leaf. So this is one important step. After this, take the leaf carefully from the test tube by using a brush and spread the leaf in petri dish, okay. You can see here the petri dish. This is called petri dish, okay. So the leaf is placed in a petri dish. After that, add few drops of iodine or betadine solution on it, okay. You can see here the iodine solution is being added here. The leaf is kept in a petri dish and iodine solution is added, okay. So after addition of iodine solution, we will notice some changes. The presence of starch will indicate, okay, the blue black color, okay. So observe the leaf after addition of this iodine solution, we find, okay, blue black color, and this presence of this blue black color is indicating the presence of starch in the leaf, okay. In the earlier concepts we discussed, during photosynthesis carbohydrates are stored in the form of starch, okay. Carbohydrates are stored in the form of starch, okay, stored carbohydrates, okay. So glucose is synthesized during the photosynthesis. Excess glucose is converted into starch. It is stored in the leaf, and this is how iodine test is performed to identify the starch in the leaves. So quickly what we have to do, we have to take a leaf that is soft, thin and well exposed to sunlight. After that we have to take a methylated spirit in a test tube. Now this leaf is boiled first in water, then in methylated spirit, okay, and when it is boiled, what happens? The green color of the leaf is lost, the chlorophyll is dissolved in the methylated spirit. After that we have to take the leaf with the help of brush, keep it in a petri dish and then add iodine solution, okay. When iodine is added, the color changes to blue black color, and the blue black color indicates the presence of starch in the leaf. So this is a test for the presence of starch in leaf, okay. Now moving to the second activity, that is carbon dioxide is necessary for photosynthesis. This is also called Mohls' half-leaf experiment, okay. So this experiment explains that carbon dioxide is necessary for photosynthesis, okay. Here take a destarched plant, okay. For destarching, what we have to do, okay, we need to keep the plant, okay, in dark room, okay. This for 3 days. If the plant is kept in dark room for 3 days, okay, the stored starch will be removed, okay. So that is how the plant is destarched. And now this destarched plant can be utilized for the experiment, okay. By placing the plant in dark and the starch is removed from the leaves. Now arrange the apparatus as shown. This is a potted plant, okay, and here you can see a wide mouthed bottle is taken, and here the split cork is inserted into that, and the leaf of the potted plant is inserted into this bottle, okay. So let's discuss about this activity. Take a wide mouthed transparent bottle. You can see in this picture, this is a wide mouthed transparent bottle, okay. After that, put potassium hydroxide KOH pellets or solution in the bottle. So inside this bottle you can see here, this is KOH. So KOH solution or pellets can be added into this wide mouthed bottle, transparent bottle, okay. After adding KOH, okay, now what is the purpose of adding KOH? This is very important concept, okay. This KOH absorbs carbon dioxide, very important statement, okay. The purpose, the objective of adding KOH solution or pellets into this wide mouthed transparent bottle is that this KOH absorbs carbon dioxide, okay. Now after that, insert splitted cork in the mouth of the bottle. You can see here, this is the mouth of the bottle and the splitted cork is inserted, okay. So splitted cork is inserted here. Now after that, we have to insert one of the leaf of destarched plant into transparent bottle containing potassium hydroxide pellets or potassium hydroxide solution. So we have to insert one of the leaf. You can see here, this leaf of a destarched plant is inserted into the bottle, okay. What happens, okay, leave the plant in sunlight. So if the plant is kept in sunlight, the photosynthesis will occur and the starch formation takes place again. Already the plant that we have used for this experiment is a destarched plant. It means starch is absent in the leaves. Now once if it is placed in the sunlight, again starch formation will occur. Now what have to be done? After few hours, test this leaf and any other leaf of this plant for the presence of starch. We have to test now for the presence of starch, okay. The leaf part which was exposed to atmospheric air and light becomes bluish black, okay. So here you can see only half of the leaf is inserted. Only half of the leaf is inserted. This half is exposed to light. This is exposed to light, okay, and this half is inserted into the bottle, okay. Now so you can see what happens. the leaf part which was exposed to the atmospheric air and light becomes bluish black, because that part of the leaf, this part of the leaf, it performs photosynthesis, because from the air it gets carbon dioxide and light is available from the sun. Okay, so it can perform photosynthesis, and here the starch formation take place. Okay, but the other part of this leaf which is placed inside the bottle, okay, where KOH solution is present, this part cannot perform photosynthesis because there is a KOH solution which absorbs carbon dioxide. So the carbon dioxide present in this bottle is absorbed by the KOH solution. So this part of the leaf do not get carbon dioxide. In the absence of carbon dioxide, it do not perform photosynthesis. Okay, as the photosynthesis is not performed by this part of the leaf, the starch formation do not occur. Okay, as the starch formation do not occur, the blue black color cannot be seen in the part which is inside the bottle. Okay, the remaining half of the leaf inside the bottle containing potassium hydroxide, which absorbs carbon dioxide in the bottle, remain colorless. Okay, you can see this is blue black color due to the presence of starch, and this is colorless because starch is not there. Okay, so this is how, okay, this activity explains the importance of carbon dioxide for photosynthesis process. Carbon dioxide is necessary for photosynthesis. This is also called Mohls half-leaf experiment. Okay, now moving to the next one. After this, sunlight is necessary to form starch in green plants. Okay, that is black paper experiment. Okay, for fix the paper, we have to take a potted plant. Okay, and now fix a paper on the leaf in such a manner that light does not enter the dark part. So this is the dark part. So this dark part does not receive the light, but the other part, this one, will receive the light. So this is how we have to arrange the apparatus. Okay, now here we can see, place this potted plant in the sunlight. Okay, after few hours of exposure to bright sunlight, test the leaf which is covered by the black paper for the presence of starch. Okay, it will be observed that only the parts of the leaf which could get light through the cut out design turns bluish black, showing the presence of starch. Okay, so here if we look in this design, if I draw here, it will be like this. Okay, this was the design which was, okay, cut in the leaf. Okay, so only this this is a black colored part. Okay, this is a black colored design, and this is not the black. This is receiving the light. This part is receives. Okay, it is receiving light. Okay, and the part in this region, okay, do not receive light. Okay, so this is how the parts which are exposed to sunlight, this part of the leaf, okay, this part of the leaf is exposed to sunlight, so it is receiving the light, and the part which are covered by this black paper, okay, then they are not receiving the sunlight. So black paper experiment. Okay, so here those parts which are exposed to sunlight will perform photosynthesis, and they can synthesize starch, and when iodine test is performed, blue black color is seen only in this region. Okay, but the parts which are not exposed to sunlight, photosynthesis do not occur because they are not receiving the light, as they are covered with the black paper. Okay, as they are not receiving the sunlight, they do not perform photosynthesis. Starch is not synthesized. Okay, and as a result, when iodine test is performed, it will remain as colorless. Blue black color is not seen. Okay, so this is how sunlight is necessary to form starch in green plants. Okay, this is all about the black paper experiment. So what are the steps? First, we have to take a destarched plant, cover one of the leaf with this black paper with a design on it. Okay, and it is kept in the sunlight for few hours. After few hours, iodine test is performed. The parts which are exposed to sunlight will perform photosynthesis, and the parts which are not exposed to sunlight, they do not perform photosynthesis. And when iodine test is performed, these parts will give blue-black color. Okay, so this is all about the black paper experiment. Okay, so today I discussed about 3 activities. One, that is presence of starch in leaves. Okay, iodine test is performed to identify the starch in the leaves. After that, in the second experiment, carbon dioxide is necessary for photosynthesis, that is Mohls half-leaf experiment we discussed. And in the third, we discussed about sunlight is necessary to form starch in green plants or black paper experiment. Okay, other activities will be discussed in the next session. This is all about today's session. If you are new to my channel, subscribe for more sessions. If you liked it, share it with your friends. I'll be back with more sessions.